Recording now.
Cool. Okay, so May 22nd, welcome everybody to another happy hour. I'm Judy Wismer, and we have Randall. You want to say hi?
Hi, again.
From Iowa State, and we've got Dustin Scholl and Luke Norris. Jessica might be coming on later. We might have Sheldon coming on and Courtney coming on. So lots of people from UNL E-Lab coming in and out as well. The reminders, I was going to ask Randall to send out the email list again through the chat box, just so new people can get on that. That's the email that if you have updates or questions, you can reach us, specifically regarding happy hour questions or comments, feedbacks.
Yeah, I'll go ahead and add that right now.
Awesome. Then I was also going to ask Dustin to post the recording from last week so folks can see that if they have it. We are currently working on trying to get these recordings archived on a website or somewhere you can look at the old ones. Because of the Beekill, things have kind of been a little bit kind of hectic in our lab. So we'll work on trying to find a good platform or a place for those recordings. So Randall, I didn't know if you had any updates or questions. I didn't really want to talk about the Beekill till a little bit later because it's a really kind of sad topic, and I've got some videos and some pictures as well. So I thought you could start, with some.
Yeah, the one thing that I have is we worked with our extension video team here to produce a couple new videos. One of them is on swarming. Well, not on swarming. It's on splitting hives and combining hives. It's good. I could share it. It's about 5 minutes if people want to see it, just as a follow-up from some of our previous conversations from the past couple weeks. Should I do that?
That would be great.
Okay. It's always awkward watching yourself, so I'm sorry if it's cringy. All right. Okay. In this video, you'll learn about splitting and combining honeybee hives. Splitting and combining hives is something that every beekeeper has to do. And there's a lot of great reasons to do it. It's a great way to manage your hive population. It's a great way to increase or decrease the number of hives that you have in your apiary. And it's also a good way to control for swarming. Splitting a hive refers to the process of dividing a larger hive into two smaller hives. Combining a hive refers to the process of taking two smaller hives and combining them into a single larger hive. When is a good time to split? A beekeeper is going to want to split a hive when that population is really booming.
In a walkaway split, you may come to a large colony, and in your colony, what you're going to want to do is make sure that in each of your deeps, you have sign of the queen by looking for eggs, young larvae, and capped brood. It's important to have eggs in each of the deeps so that the colony can successfully requeen itself. Take your top half of the colony and introduce it to a new bottom board. And then you can keep your lid on this colony, and then in your home colony, you're going to want to place a new lid on there. And the assumption is, since we've checked for eggs and young larvae in each of these colonies, one of them is going to have the queen in it, and that colony will continue as normal. And then the other one, you're assuming, is going to requeen itself with the eggs present in that colony. It's important to make sure when you do your walkaway split that you take one of your colonies and you move it to the other side of your apiary so that the bees from that colony don't drift back into the home colony. Then you can just walk away.
One of the disadvantages of a walkaway split is that it can take some time for the hive to requeen itself. Usually a queen will start laying eggs at about 3 to 4 weeks after the split happens. Another method is an easy split with a queen excluder. Utilizing this method, you actually place a new queen in a cage inside the hive at the time of the split. And that way you can get that population booming faster. To do an easy split with a queen excluder, I'll go out to my apiary and I'll bring a hive body that's full of empty frames. Drawn comb that doesn't have much in it, maybe a little bit of nectar, but mostly empty. This is going to be the receiver hive. I'm going to go to the colony that I've decided to split, and I'm going to look through the frames to look for brood. Not just capped brood, but also open brood with eggs and larvae in it. I want to remove 2 or 3 good brood frames out of this hive. And I want to be sure that I'm not taking all of the brood frames out, because I want this population to still thrive. As I'm removing brood frames, I'll gently shake the bees back into the colony. That way I can ensure that I'm not taking the queen with me and putting her in this new one. I'll remove 2 or 3 of those brood frames with the intention of putting them in the receiver colony. Then I will take 3 of the empty frames and place them inside the mother colony. Now the colony that I've chosen to split is already strong, so I know that the queen will immediately start laying eggs in some of those empty cells. Now I have my receiver colony that has some brood frames in it, and I'm sure that the queen's not in here because I shook the bees off inside the first colony. I'm gonna place the queen excluder on top, and I'm gonna place my new receiver colony on top of that. I'm gonna close up the hive, and then I'll return in a few hours or the next day. In that time period, this heap is gonna fill up with bees, but not the queen, because the queen excluder is in place. At that point, you'll be able to return, remove the receiver colony, and place it on its own bottom board. Now I've split 1 hive into 2 hives, but this hive still needs a queen. So I'm gonna take my queen in her cage and place her in here. Now remember, you can't place a queen directly into a colony or the colony might reject it. They need time to get used to her pheromones. Now I'm going to move the receiver colony to the other side of my apiary.
The farther away you can get the colony, the better, but I would aim to go at least 10 feet away from your home yard colony.
With the mother colony, don't forget to remove the queen excluder and put the lid back on. And a few days later, I'm going to come back and check on the progress of the queen and make sure that the hive's accepted her. Keep an eye on both the hives over the next few weeks and make sure that they're healthy and growing. That's how you do an easy split with a queen excluder.
You're going to want to combine your colony if you have a colony that's very weak or a little bit unhealthy. If your colony is performing poorly, not collecting a lot of pollen, is also not collecting a lot of honey, or the queen's not performing well, this may be a good time for you to combine that colony with another weak colony in your apiary. If you have a colony that has a queen present in it, you're going to want to take that queen out. If your colony is queenless, you can move on to the next step. Here we have a queenless colony that we want to combine with our stronger colony. We can go ahead and crack the top of our stronger colony. We can provide them with a quick puff of smoke just to calm them down.
Make sure that they're off the edges.
Get them off the edges of the colony. We're then going to take our newspaper and lay this over the top of the colony, gently.
And then I'm going to come in with the second colony.
With our weaker colony going on top. It's going to take 2 or 3 days for the bees to eat through this paper, allowing ample time for the pheromones from this colony to merge with this colony so that the queenless colony on top will accept the queen at the bottom.
That's combining a hive.
Nice job. You can combine your colonies anytime during the season if you have several weak colonies that you want to give an extra chance to survive. But the most common time for combining colonies is actually in the late fall prior to going into the winter. The reason for this is that you want all your colonies to be strong enough to have a nice large cluster of bees in order to survive through the winter. So taking your colonies and making 1 colony or 2 colonies into one nice large strong colony can help them have a better chance of getting through the winter.
Thanks for watching this video. We hope that you learned something. And if you have any questions, please feel free to leave them in the comments below.
Oops. All right, that was you're the first people to see that, so
Great.
any feedback or anything like that is absolutely welcome. But
Nice shots of the beard.
I miss it. It was a lot bigger then.
Yeah, no, I like it. I thought it was really well done.
Yeah, it turned out okay. It was interesting to shoot, to film, but it ended up, it feels very like HGTV, but yeah. So yeah, those are our recommendations for splitting and combining.
Yeah, and I like the use the demonstration of the queen excluder because we don't really show that one. We often will have people split, and oftentimes we forget that sometimes you really need to be able to find your queen. So if you can't find your queen, there's different ways to do it. The queen excluder is definitely one way we've shown people to put the queen excluder in and then come back to see where the eggs are. And then you know where the queen is, and then you know which one is your mother and which one's your receiver.
Yeah.
Yeah.
Yeah, and then there's another one, and maybe I can post the links here in the chat. There's a second video that we did that's about, oh no, what is it about? It is about requeening a hive. Yeah, it's about requeening. So I'll just post the links to those two in the chat right now.
Thank you. Do we have any questions? All right, so if you do, please feel free to put it in the chat. I will, Dustin will continue, we'll all just continue monitoring it. And then I'll kind of go over this dead bee kill thing that's happening. So this has been something that's happened consistently with us for a while now. So I began my position in October of 2016. So spring of 17 was the first time we put colonies out in this property, and we had dead outs. We saw bees out front, but we were kind of caught off guard at this location. So the following year, this is 2018, we equipped colonies with dead bee traps, and that is what I'm showing here. And these were on 3 to 4 hives at 3 locations in this research farm at UNL. And so this is 1 week later. This is the acute bee kill that's happening right now. We had these bees tested for pesticides, but one of the issues with pesticide testing is you kind of have to know what you want tested for in order to see if you can find it. So if you send these samples to the USDA lab and they say they can screen for 170 different kinds of compounds, there is a very specific list of those 170 compounds that they're looking for because they have standards to compare them with. So unfortunately, whatever those standard list of compounds were, there wasn't a whole lot that came out that would explain these huge losses of bees. This is the map to kind of show you. This is our honey house, our insect lab, and then another organic cover crop study that we're doing over here. So hives were at these locations. So each of them were probably less than a mile apart from one another. And this is part of a pesticide drift barrier study that one of our PhD students is doing. So here at this insect lab, there's a little corner of the property where there's a nice strip of trees. So we planted pollinator habitat in 17 different locations near tree lines and not near tree lines to see if we can mitigate some of the drift from neonicotinoid seed plants going across the landscape. So when you plant corn or soybeans, but it's mostly corn, when corn is planted and that corn has seed treatments on it or seed treated coatings, those have chemicals that have usually an insecticide and several fungicides. And the issue with the insecticide is that this chemical tends to be very highly toxic to insects, and it can be taken up by the plant and move throughout the plant. So it's systemic. It can be taken up by the root system, be translocated and be expressed in the leaves, and then also in the nectar and the pollen. So that's kind of where we've been really interested in trying to figure out, well, can we mitigate or can we kind of reduce exposure from this dust that comes from the seed treatment during planting? Can we safeguard the bees by protecting them by just altering where we put them on a farmscape? So that experiment is happening right now. And each year we have these massive die-offs at this one location. So again, this year we Last year, we deployed the hives in July, thinking that we could avoid exposure because those colonies died in June and July. And so we thought, well, let's deploy them in July and see if we can avoid exposure. We still saw losses in July and then continued every month or every week throughout the season. This year, we deployed them, not the first week of May, we deployed them May 12th. But here, Luke went out and collected it this Monday. That was the image that we saw, the traps from Monday. And then this was 24 hours later. I went out on Thursday, and this is what it's looking like right now. So again, on Monday, those traps were cleaned out. And so again, Thursday, these traps were cleaned out. The bees are collected, they're in the freezer, they're waiting to be tested. This is a video. I want to show you what those bees look like when they come out and they're dying. So sometimes you can see young bees in there. You can see bees that are kind of hunched over and they seem to have neurological issues. Some you can actually see trembling. Some will be upside down and you see their legs are moving and their wings are twitching. Trembling, twitching, these are all symptomatic of a poison that targets the central nervous system. And many of these insecticides are targeting central nervous system. So they're nerve poisons. So you'll see some of that trembling and disorientation. There's 3 traps on this palette. So this is just to show you that when you don't have a dead bee trap here, it's very difficult to see or gauge the severity of the losses. So this is that 4th beehive or the colony that does not have a dead bee trap. And I don't know, it appears to be losing at, just as many bees as the other ones. I looked at the hives on Thursday. They're all queen right, amazingly, but they were very big going onto this property. And there is strong evidence of spotty brood. So sometimes it's also referred to as the crud or cruddy brood. On the right, you can see a drone frame of pupating bees that have been uncapped and they're all dried up and they're kind of, these tongues sticking out. Let's use my annotation tool here. So I'm talking about these things right here. These were capped pupae that seem to have been uncapped here or in the middle of development and they're getting discarded. Here you see just really shotgun brood pattern, lots of holes missing out of the brood. That's very characteristic of something going on that's impacting the brood. Let's see. If I zoom into this picture, beekeepers are always asking me what are some of the signs that I should look out for in terms of pesticides or brood issues. You guys, some of you know where the annotation tool is. Randall and Dustin, you just figured out how to use that too. Or Dustin just figured out how to do it. Can you guys mark or highlight cells that you would suspect as having issues? Like signs that there's something wrong with the brood. I can't hear anybody. Oops, can I see anybody? Oh, sorry, I can't see, okay. I was trying to change the view and I totally changed everything. Okay, there we go.
Can you, yeah, there we go.
Does anybody see what some problems? Yeah, see that larva, someone just circled a larva that's kind of swooped down in its cell. The next marking is a discolored larva. And then the next markings are definitely something that looks starting like it's dried up and starting to die. Yeah. Awesome. Okay, you guys ready for another one? I'm trying to make this interactive and fun, even though this is a horrible, horrible thing happening right now.
So those problems, are those because of the pesticide or because there's a problem in the hive?
That is a great and insightful question. So I wanted to bring this up because this is an indicator that something is happening to the brood. I still can't really tell yet, and we can talk about this more, but this certainly is indicating that something is impacting this brood. Here's another picture and see what you can kind of highlight as suspicious indicators that there's something going on with the bees. Yep, there's one right there. Swooping down larvae again. Yeah, and that one looks like it's like untwisting, almost characteristic of EFB. Some of them look like they're just sliding down. Some of them look like they're elongating. Someone just circled a perforated cell. So these are openings in the cell. So that might be indicating that bee is getting, some bee is starting to hygienically remove the larvae. And so that's evidence of hygienic removal. Sometimes you see perforated cells as a symptom of high mite load, or other diseases can sometimes have sunken cells like that too. But we know that the mite loads and this colony was healthy going in. All right, next one. This one I think is the last one of this set, but what do you see here? Yeah. The other thing, you know, as people are marking where these disease cells are, think about where, what you're not seeing in these cells as well. What do you notice that you're not seeing? I have one too. These ones also, I'm in pink. Oh, maybe I'll make it bigger. These ones also are kind of like these scales of these dead broods. So sometimes they're even harder to see because you just kind of see the segments of the larva, but it's dried up and it's kind of dead. Notice, yeah, let's see if I can get rid of this. Oops, I didn't mean to save that screen. Notice, I'm going to show you this video and you can chat, enter what you think is missing from these frames. Notice how erratic and shaky the bees are moving. They seem kind of like they're flickering, they're trembling. If they were to fall on their back, some of them have a hard time righting themselves back up. Also, notice how the brood is all fifth larvae brood. There's very little young brood, and there's very little resources under that brood. You want to see that again, but specifically looking at the brood. It's very spotty. There's not a whole lot of food. There is some one-day-old larvae in there, but what we're missing is like the little, you know, everything in between 1 day old and 5 day old.
Judy,
So,
I have a question. Was this hive aggressive at all? Were they?
No.
No.
Mm.
Did it sound? Did it sound weak? Did it sound off? Or are there any other signs other than?
It looked like it had more brood than adults. So these colonies went in and with, you know, 20 plus frames of bees and then maybe 12 to 15 frames of brood. They were just packed. And so this frame right here is really beautiful. What do you guys think is going on here? Like I started seeing a lot of these cruddy brood frames, but then I was also seeing all these really pretty sealed brood frames as well. So I've got this little investigative icon because this is how I think when I'm in the field and I'm trying to troubleshoot pesticide exposure incidences. The bees on this frame are emerging. So if we look really close, there's evidence here of bees chewing their way out. There's evidence here of bees that have already chewed their way out. So all of these are chewed out bee cells. All of these are newly emerged bees. You can see how fuzzy and light colored they are. So this frame tells me that these bees are emerging today. And so if we back calculate, if they're emerging on day 21, that means that brood was capped by May 9th. We deployed the hives on May 12th, meaning when we brought them on location, they were already protected in their seal cappings. So there was no introduction of whatever contaminated food source or other means to harm these bees. So they look beautiful and they're starting to emerge. But then in the dead bee traps, you start to see just adults of all ages. So what I tend to think about is, okay, something occurred between May 12th and the 21st is when we first noticed it. So within a week span, we notice die-off and losses. We know that there's eggs present, but we don't see a whole lot of young larvae. We know that bees can sometimes cannibalize young if they don't have enough pollen, and then they basically shunt those resources to the older brood so they can quick and go into pupation, so they don't actually cost any more energy to produce. So it makes sense to eat, some of the young larvae that you can't take care of, to shunt resources to the ones that can make it to adulthood. So there's two things that might be happening here. Either the bees are getting into something in the environment and the workers are not coming home, the foragers, and there's not enough food to take care of the brood, in which case the brood will still look cruddy. They'll still look like they're coming out of the cells begging for food. So you'll still see brood dying, but it may not be associated with a disease. It may be associated with starvation. Or they're actually getting fed, but that food is contaminated and there's a direct effect of that contaminated food source. So there's a few things that could be happening here. But I mentioned those two things because when you get a bee kill investigated, sometimes you get resources and you take the samples in the hive, and if you find not a whole lot of pesticides, it may not mean that the pesticides aren't causing something. Maybe that all your older foragers aren't coming back to the hive, and all the hive bees that are in the hive are just eating up the resources that are present and starving. I don't have a whole lot to tell you in terms of what is happening. We are taking samples. We're working with NDA, Nebraska Department of Ag. We're working with EPA. What I encourage beekeepers to do is to get some dead bee traps and to, if they suspect pesticide issues, to put them out to help us identify seasonal loss patterns. When are these bees getting exposed? Where are these impacts occurring? How are they occurring? And what compounds are we trying to assess? Because we're finding that with these dead bee traps, we're able to triangulate and isolate some potential sources of contamination. So these are some of the citizen scientists that are working with us from last year and the year before. Kansas and Iowa had some really low losses, so that was awesome. You'll see our NREC site in comparison to all the other traps that we had going had extremely high losses. We just look at the UNL traps. This is the average dead bees. We had 3 different sites. This is really interesting. The one in orange, those orange lines were 3 traps in an orchard. And that was in Nebraska City. So these aren't a lot of bees. I mean, a few hundred bees is not a whole lot, but it's interesting because we think it coincides with potentially early fungicide spraying, but we don't know for sure. But then you see these green lines. Those ones are the traps at the UNL campus. And so those losses coincide much later than the orchard bees. And we think that there might be something going on either on campus with spraying or mosquito controlling in the city. We're still looking at potential sources of when that's happening. But you can see when we narrow down the window, it's easier for us to track down point sources. So this blue one is what we're working on right now, and we're very close to trying to figure out the potential cause of these bees. I can't, since we're currently in the process of investigating, we can't divulge more than that. But I do want to show people that at this same site, we have this PhD student who's looking at wild bees. And when we see increases in honeybees, we also see decreases in wild bees on the site. So, and that also includes the flies and the beetles and butterflies. I think the way we investigate pesticide kills is all wrong. It's pointless to kind of retroactively try to figure out what killed them. I think what I'm trying to encourage beekeepers to think about is to proactively monitor for issues. Right now, these dead bee traps are helping us kind of identify potential practices that may have led to these harmful exposures. So we're in the talks with EPA to figure this out right now. But I really, really hope that beekeepers, this is easy to make. And this is just like we monitor for queen health, we monitor for brood health. This is an easy way to kind of just get more information about your hives. So other beekeepers that have started implementing the bee traps in their operations have found these very useful. So does anybody know what's wrong with these bees found in the traps? Where did my chat box go?
You can either unmute yourself or put it in chat. This is the interactive part.
Oh yeah, yes, please. If you can either unmute yourself or put your thing on chat, but do you guys see anything wrong with these bees that are found in the trap?
Eric P says no wings. Robert Burns says too young. And Patrick says, can you go back to the instructions on the dead bee traps again?
Oh yeah, sure.
Maybe COVID.
Yep, absolutely. So, and we can post this too. I think it's on one of the websites, GPMB website. But yes, those bees are too young. They're young bees. They're nurse bees. So we've been teaching the beekeepers how to diagnose the bees that are coming out of these traps. So if you see young bees coming out of the traps, that might indicate that those nurse bees might have consumed some contaminated pollen, because they're the only ones in the hives that are disproportionately consuming large amounts of pollen. So if you think about it, if there's contaminated nectar and pollen and the bees are out in the field, they consume contaminated nectar, they probably won't come back. But if it's contaminated pollen, they're not consuming it, they're packing it on their legs and they're bringing it back to the hive. So the bees that are consuming it are potentially your nurse bees. If you lose all your nurse bees in your hives, then there's nobody to take care of your brood, there's no food, then you start seeing brood diseases and brood problems. So I think that is what's happening here, is potentially there's not enough food coming back to the hive and there's not enough workers to take care of the brood. So what I would do to mitigate this issue would be to feed pollen supplements, which is what I've done. And if I had other hives that weren't impacted, I would put more frames of sealed brood for them so that they have more nurse bees. Yeah. So here's another picture. These ones are deformed wing. So a lot of beekeepers who thought they had a pesticide issue have come to realize that no, really, my Varroa treatment has been ineffective this whole time, and it's not pesticides, it's Varroa that's killing my hives. So there's been a couple of beekeepers that have been doing the OA vapor, but have been doing it incorrectly and not plugging it thoroughly. And so they thought that they had the Varroa mite under control, but using the dead bee traps, they were able to actually see lots of deformed wing and actually mites when they collected the dead bees. They just piled on the bottom of the bag. So that's been really cool. And then we did receive this question from somebody who said, well, you know, my bees disappeared. I installed them, and this is what I have left. What do I do? That's kind of how I feel like our colonies are. There's a queen. There's still queen, right? But there's very few workers left, and it's starting to really dwindle. In this case, we would stock this frame with more brood frames of sealed brood, ones that are going to emerge very quickly to give this struggling hive some workers. I don't know.
Judy, you want to talk about a little bit if like Chris is asking, how long do you put them out and stuff? But for a normal like backyard beekeeper, if you go out, if you deploy a dead bee trap, like what's normal loss versus heavy loss? Like, can you talk about that? Do you have any photos or anything like that?
Oh yeah, normal loss versus, yeah, that's a great question. You know, one of the things that we're trying to do with the dead bee trap study, and this is that citizen science study, is to track what we call abnormal mortality. So basically, everybody's going to have a baseline mortality that they just lose a certain percent of their bees each week, and for some of these. I guess I had one that is probably being hidden, but for some of these, oh, I don't have it anymore, darn. For some of these traps, the weekly mortality was like 10 bees or 20 bees. In some of the areas, like some of our partners in Kansas, maybe their weekly average was 40 or 50 bees. In our NREC or our orchard site, it was about 100 bees, a week for 4 weeks or something like that. So it's not really like a certain threshold. It really is dependent on how big your colonies are, and respective to how large your population is, how many of those bees are getting lost or dying. The traps are not catching 100% of the dead bees. They're catching a pretty low percentage, maybe 20 or 30%. They're much more effective in the spring when the colonies are smaller and they don't have enough what are they called? The grave digger bees. Undertaker, thank you. Not grave robbers. The undertaking bees to throw them out. So we see that the traps are less effective when the colony is really big. I think they just have more workers to take them out further. The point is to just monitor normal mortality, and then when you see upticks, that's when you should be more careful at maybe doing a more thorough inspection of your hive. Like if I see an uptick in mortality, I probably won't just do a quick check. I might do an inspection, Yeah. And then every time, like Dustin said, every time you check your traps, you empty them so you know what's new. If you do see a consistent loss, or if you start to have problems, like the spills that you're seeing coming out of our hives are probably in the thousands. So these are not just a few hundred bees. These are 1 cup or 1/2 cup is about 300 bees. you can visually imagine, you know, there's several cups of bees, there's several thousands of
You guys
individuals.
want any indication, and Luke can attest to this, one, it smells like there's so many dead bees, it smells like a dead animal. And when Luke went and picked them up the other day, he filled a gallon baggie half full, like from each of the traps. Like that's how many dead bees that we're having come out of these. So it's a lot, and it's really sad.
Mhm. Mhm.
Judy, I wanted to ask, so for like just beekeepers in general, if they do suspect just you know, that it is a pesticide, is there a good way for them to verify that for themselves to find out you know, that it is in fact a pesticide?
You know, that is a great question. And I am finding this process extremely frustrating. So our program is really focused on trying to help beekeepers with these pesticide issues. And so we've come to a point where we have hit a dead end and requested help from NDA and EPA. And that process has been extremely slow and frustrating. So while that regulatory process is going through, I recommend that beekeepers become proactive and put those traps on, record, get a hold of us so we can kind of help guide the data collection. And then you can monitor that because you know, this started Monday and I'm told that we will meet sometime next week to talk about this. So, or the week after. So, you know, like in the meantime, I'm collecting bees, I'm collecting samples, I'm recording the health, I'm monitoring what I can. And I certainly think that there is something going on in Nebraska where if there are issues, please let us know, please set up these traps, please, you know, we want to try to isolate these point source contaminations. So I do have a Kansas, I had a few traps from Kansas in 2018, and these were different beekeepers, but they all had upticks in losses at the same time. So they were, I think, within 20 miles from one another, but this helps us triangulate potential areas that we can further look into what's causing this exposure.
Well, and in, I don't know if anyone tuning in is from Iowa, but in Iowa, since we have the Iowa bee rule, which says that beekeepers can receive compensation, well, can basically, it's illegal to spray a flowering crop during the day if it's within a mile of an apiary, a registered apiary. So if people suspect that there has been a spray that's caused a dead out on their hives, they can contact the pesticide bureau, the Iowa Pesticide Bureau, and they'll send someone out to test.
We
Yeah,
have a question, and then Judy, I've got a little slideshow with some questions about the Nitadulit.
Yes, please do that. And then Chris also had an image to share too.
Yeah, Chris says, I have a hive that doesn't seem to be growing. I have a couple of pics that he can share to get your thoughts on it. It was a nuke and hasn't done much at all. No new comb, but it has larvae and nectar. Let me know your thoughts and if you'd like me to share the pictures.
How much sealed brood and brood was in the nuke when you received it?
You know, there was only there was a total of 4 actual frames. One of them was empty, so really had probably a couple of frames of brood. And it really hasn't grown much. I don't see anything. It's in a 10-frame hive box right now, but they haven't done anything to make any comb.
Do they have food and nectar and pollen and things?
Yeah, I see pollen in where they're at right now. It's a real old comb that they were in to begin with, and it was real tight, and it's still tight. And I keep thinking, well, I wonder if I should separate some of that out and give them some room. But I just took off when I first got them. Probably 3 weeks ago, I had sugar water in there for them, and I took that away this week because the other hive that's growing, they didn't need it. They were bringing in pollen and nectar. And so this one, I thought, well, it's been 3 weeks. They should have plenty to get out themselves.
Yeah, I mean, let's take a look at the pictures. The first thing that pops in my head is how much of the young bees you have in that colony. Well,
Okay.
Randall, what do you think?
Yeah. I want to. Let's see what the pictures look like.
Let's see. Can you see that?
Mm-hmm.
And then let me pull up another one for you. And there's one more. So that's what it looked like when I got in. When I
I'm
got
still
them.
only seeing the first one.
Okay.
Still don't see it. Judy, are you seeing it, or are you seeing the same photo?
I see the same photo. Yeah.
Oh, well, let me try the next one. Maybe I have to share each one.
Yeah,
That's
and
it.
you might have to share new if you go to the view, to the options.
Okay. I'm going to pull that one off. Can you see that one now? Well, let's see. New share. Here we go. Sorry. See that one?
No.
Yep.
There we go.
Oh yeah. Yep.
Do you have any images of like
Full
larger,
frame?
like the frames? Yeah.
No, I should have done that.
Because
Sure.
I mean, I don't really see a whole lot of young bees. They do seem older, but it's hard to say. There's not many bees there.
No, there weren't a lot when I got it either. This is what I got, and see if you can see the hive box. But it looks just like when I got it. No different.
Oh, let me ask you something. Are the 2 frames on the outside full of food, and is it capped?
No, I mean these here.
Yeah.
No, there's no food in them. They're just empty old comb on them. On the inside here, there's some food in there and on here, but there's really not much going on the outsides at all.
Mm-hmm. So when we were doing the experiment with the waxing project and we put in the old comb and put food on the outside and then brood on the inside and the foundation on the outside, they didn't draw out any comb until we moved the foundation 1 frame in. So taking that new comb and putting it in between the outer and the, bringing it 1 frame in on both sides.
So don't put the foundation on the edges. Bring the foundation in. Okay.
Well, once it warms up and you don't have any kind of a worry for brood chill, then you start to slowly checkerboard it inward. So move 1 frame in, let them draw it out, and then you can continue to kind of do that, working them, kind of stimulating the wax production.
Yeah, I've got another hive that is, drawing out comb. And so I thought, well, maybe I'd pull one of those out and put it in this one and see if that helps them.
Yeah. Yeah, I mean, it looks pretty small. So I would also, if you have another colony that's doing really well, give it a frame of sealed brood that's going to emerge. And maybe it just needs, it could be that you just have some really old bees there that aren't great at wax producing or not the great age for that. So a stimulation of some young bees might help.
Okay, I'll give it a try. Thanks.
Do you have any excess wax, Chris, that you could?
I have some wax, some of those wax cubes you get, from the places where you get all your equipment. And so I could, use that and paint it on the combs. Is that what you're thinking?
Yeah. Just they'll take to it better, but yeah, I'd agree, like with Judy and Randall. First, introduce some frames of cat brood to get some young bees in there that will grow up a little bit and draw some wax for you. I bet you got a whole bunch of older bees on this, and they're just hanging out.
Yeah, that's what I kind of think, and probably the queen might not be that great either. Okay,
Good.
thank you.
we got another question in the chat. What temp causes brood chill?
Oh, that's a good question. I think it has, well, I think it varies, right? So a lot of times we talk about how we don't really want to open up hives below 50 degrees because they don't really fly, and you know, it's not good to expose them, open up that warm air, pocket of air. I don't know if there is a set temperature that causes brood chill, because it kind of feels like it depends on the colony size. Because there's going to be like really strong colonies that can generate heat and do really well with keeping their brood warm. But then there's colonies that maybe they don't have enough workers and they have too much brood. And when they cluster up, there's brood that hang out on the outside and those get chilled. So it kind of depends on how cold it gets and how big that population that causes them to cluster and kind of move away from the brood. I don't know. That's Randall, do you have?
I think that's a great answer. I think brood chill doesn't have so much to do with the I mean, it has some to do with the outside temperature, but it's saying more about the colony's size. If you get in there and you find that it's cold outside and your colony's not big enough to keep all of its brood warm. Yeah.
Also, maybe a little bit to at what age your brood is, right? Like eggs versus cat brood. Cat brood can definitely like if you're transferring, if you need to transfer a frame of brood to another apiary, you know, typically you do that with cat brood. If you have to, like if that's a must, you wouldn't do open brood.
Right, and in the winter, I mean, when you're looking at those thermal guns, if there's no brood, they keep that hive temperature pretty cold, I think in like the high 50s or 60s. But if there's brood, they keep it at like 90 or something, pretty hot to keep incubation going. So, you know, if they're clustering tight enough to condense and they're exposing areas to that cold, then I think that's where you start to see brood chill.
Robert says brood nest temps are in the low to mid 90s, so enough bees to keep warm.
Yeah, thanks. Yeah.
Okay, you ready for my presentation?
Yeah, do it. These are questions from email, right?
Yes, these are questions that I got a lot of these this week. I got them personal Facebook messages, emails, Facebook groups. So what does everybody think this is?
Cool, we got 3 minutes.
A mite?
A mite? So maybe a few more of these will help. Like what in the chat, what does everybody think they are? Think they're small hive beetles?
Yeah, that's what people are saying. They some kind of beetle, not small hive beetle.
I realize that's an ant in that picture. So here we have small hive beetles at the top, and we have sap beetles at the bottom. And there's over, according to Minnesota's extension website, and there's over 180 different types of sap beetles. But they are all in the Nitidulidae family. So sap beetles are also referred to as picnic beetles, and they become a nuisance in gardens in late summer. Has anybody ever seen these in tomatoes?
Yes. And I think strawberries and other fruits, soft fruits. Yeah.
They are not a problem in our hives. These are longer and more oval shape than a small hive beetle. And then there's a picture of them in some bruised tomatoes. So here's your small hive beetle. That's typically has the clubbed antenna. That's a big telltale sign. And they'll be brownish, but the older they get, they're black. And they're roughly the size of a ladybug. They're native to Africa, and they were brought to the U.S. accidentally in 1996. And they can fly. That's how they get in the hive. Here's their life cycle. It starts down here in the lower left-hand corner, so they land and crawl into the hive. And then they like to avoid light, so they tend to congregate in spaces that are inaccessible to honeybees, or the honeybees will corral them and actually make a beetle jail. The females will back up to the cell and they will lay a whole bunch of eggs, and sometimes they lay it on top of pollen, or they'll puncture the cell wall. If you have a weaker colony, then the eggs will hatch in 2 to 4 days and they immediately start searching for food. They will feed on everything in your colony: pollen, honey, and bee brood and eggs.
Yeah.
When they have had their full, after 7 to 10 days, they mature and make their way out to the ground and burrow in the soil. And then they pupate into what we know as the adult small hive beetle. And then this whole process starts over again. So here's an up close image of what the larvae look like. And it's not the adults that do the damage in a hive, it's the larvae, and they do the slime out. So I know that image is a little blurry, but I made sure to have high quality gross images for us. So here's when it gets worse. Here's when the slime out starts to occur. And there's a bottom board after an aftermath of small hive beetle slime out. And that was actually, we took that picture last summer at an aphis survey. And then here is a whole bunch of small hive beetle adults and larvae together in a giant mess. Ugh. I don't want you guys to confuse small hive beetle larvae with wax moth. We have small hive beetle on the left, and we have wax moth on the right. Wax moth are always going to leave a webbing behind in the combs, and they'll have their frass or their poop in there. That's all those little black flecks. And there's a nice image of one of my colonies like 6 years ago of a complete wax moth, if you really want to know what kind of damage they can do. So prevention for small hive beetle. We want to keep strong queen-right colonies, and we recommend mechanical traps for the adults. We really like the blue unscented Swiffer pads over any of the beetle blasters or the dryer sheets. And we found with a little bit of an experiment not to use the off-brand Swiffer pads. They're actually made of a different material. And we found that the beetles get caught up in these Swiffer pads a lot easier if you're spending a little bit more money on the Swiffer brand pads. This isn't a plug for Swiffer. It's just they work better. So you can place your Swiffer pads. You can just take them out of the box and rough them up and place them on the back or the front edge in between your brood boxes. This can be done anytime, so ignore the fact that I've got an Apiguard treatment on there in fall. Personal word of warning, don't place your Swiffer pads below or above a queen excluder. You'll only make that mistake once. You'll be picking Swiffer pad out of your queen excluders all winter long. And then on the right here, we have a Swiffer pad that is full. Yes, there will be some bees that get caught in there, but it also they will corral the beetles into that Swiffer pad. They'll also put propolis all over it too, but they're a really good catch for the adult beetles. And we like these because the Swiffer pads are fluffy right away, and the bees can start corralling the beetles towards them right away. Some of the other products that are out there, the beetle towels, they take time for the bees to rough them up before that they're effective in trapping beetles. And then the beetle blaster traps or the jails require you to fill them with oil or some kind of bait for the beetles. And the bees end up propolizing them up. So we really like the Swiffer pads because they seem to start catching them right away. And pollen patties, at least here in the eastern half of Nebraska, are not needed, but if you do need to use them at some point, we do recommend that you cut the patties in quarters and only place a quarter of the pollen patty on the colony at any given time and put it directly over the cluster of bees. And if you find small hive beetle larvae in your pollen patty, dispose of it right away. And then I'll just display these references I found today, just for video purposes. And that's a little bit about sap beetles and small hive beetles.
Thanks, Dustin. We're running a little bit long, so there in the chat box, there was a question about whether or not you could put something into the soil. We have played around with entomopathogenic nematodes. So these are things that come in kind of a dry powder form that you put into a solution, and those are the nematode eggs, and then you seed it into the apiary in the lawn on the soil. So these are specific nematodes that attack, they're generalists, but they attack the pupal stage of the small hive beetle. We've been playing around with them in some of our apiaries, and some of them that did have small hive beetle anecdotally did have these nematodes seeded in the apiary, and it seemed to have been effective for the following year and two. So I don't know. We don't have any scientific basis for that, but that is an option if you find the beetles are really, really bothering you.
We've got more nematodes to put in the yards this next week. So
Where did you purchase the nematodes?
Arbico Organics.
There's a number of websites, but I would if you are interested, look for those specifically targeting small hive beetle. There are different varieties and lots of different species of nematodes. Many of them are used for these organic methods of pest control. So there's different ones that are specific to different insects.
In Iowa, small hive beetle hasn't been too big of a problem, mostly just in the southern border of the state. But each year it sounds like it's sort of like working its way up north across the state. So good, that's helpful.
Yeah, and that has to do a lot with the warming winters, because usually a cold, hard winter will kill off a lot of the pupating larvae the insects in the ground, the developing pupal stages in the ground. But yeah, it seems that if we have warmer winters, they're more likely to come out strong.
Great.
Anything else, guys?
Looks like Barb has a quick OA dribble question.
Yeah, what's the question? And it looks like Patrick just mentioned Keith Pelleplane does a lot of research on nematodes in Georgia, so there might be a resource there or two on specific species. And Jamie Ellis also does a lot of small hive beetle research, but he does it a lot more looking at the behavior of the beetles. And so he's got some really cool, interesting life history studies there.
In Nebraska.
I'm sorry, go ahead, Dustin.
Oh, and I was just going to say, in Nebraska, we seem to see a big uptick in small hive beetles in July and August. Like, it seems like that's when they get really bad. We did have one colony last week that was pretty weak, and it did have some of the larvae in a corner. So we're freezing that box right now. We had to drop the colony down to one box so that they had less room to police, so that they could take care of the larvae themselves. But the extra box we took off, we're freezing, and then we'll use it back out in our apiary. But yeah, same thing, Robert. We usually lose one or so colonies in the lab to small hive beetle slime out. It seems to happen really quick. I don't know where Barb went with her OA question.
Can you hear me?
Yeah,
Can I ask
Barb.
it verbally? Hi,
Sure.
guys. Happy Friday.
Happy
Sorry,
birthday.
So I pulled a split really late, like the night before I got a queen, and did my alcohol wash at the same time, and I needed to treat. So I didn't want to put Mite Away quick strips in, worried that the scent would, overwhelm the new queen pheromone. So I did an oxy dribble, but it still had cap brood. How soon can I repeat with another oxy dribble before I release the queen, once some of that brood emerges?
I would wait a week, I think.
I mean, you can release the queen and let her start egg laying and just, you know, make sure she's doing well and she's accepted before you do a second treatment. Yeah. You don't have to keep her in the cage.
Would you wait? I mean, would you wait longer than a week then? Let her have her 3 days, release her, and then give her a week before you redid it?
Oh yeah, I would definitely give before you put on a second treatment, I would make sure that the calf brood that was you know, was capped at the time had enough time to emerge. And two, oh, I lost my train of thought now.
You could wait up to what, 10 days after that new queen started laying, right?
Yeah, that's what it was. You want to make sure the queen is laying eggs before you add another treatment, because if something happens to the queen, then the colony can rear another one. Wow.
And, wow, good answer. And then the follow-up, would you re-cage her when you were going to dribble the second time, just for the couple hours that you dribbled to avoid hitting her, or no, not worry about it? Okay. Thanks.
She wants to say hi to Dustin really, really badly.
We all do.
Happy birthday, Barb.
Thank you.
Randall, did you have any final comments on that last question, or any other final comments before we close it up?
Nope. I'm good. Thank you, everyone, for coming.
Yeah, thanks. I mean, these have been really fun chats. So please send us more pictures and questions, and we will see you next week. And maybe I'll have some updates, hopefully good ones. But
Tell your friends. You can share this event out everywhere. So we want to see more people in here. So.
Mom, I want the light on.
All right. Well, bye, everybody.
Mom, I want the...
Thanks, everybody.
Happy Memorial Day, everybody.
Bye.
Bye, Dustin.
Goodbye. You want to see Willow?
Mhm.
Yeah? Okay, hold on.
Bestie's here.
Come here.
Oh,
There's the Willow.
Look at your hair, so wild. These are the next generation beekeepers right here, man.
Oh, oh, oh.
Oh, we got a piggy. We had a horsey earlier.
Well, thank you, everybody. And I'm going to sign off. Have a wonderful weekend.
Thank you.
Bye-bye.
Bye.
Bye. Thanks.
I want to show you something. Look,
Oh,
this light turned on.
oh beautiful.
Holy
Bye.
cow.